Hi, everyone. I just want to start by saying thank you so much for allowing me to speak during such an inspirational conference, and thank you for organizing such an inspirational conference. As a student, it's really awesome to see where the field is headed. So today I'm going to talk to you about the prebiotic inulin and how we used it to beneficially modulate the gut-brain axis and enhance metabolism in an ApoE4 mouse model. So obviously we've had a great background on the ApoE gene today. So the ApoE4 gene, as we know, is a genetic risk factor for Alzheimer's disease. It's also a risk factor for cardiovascular disease. And we want to take a little bit of a different focus here. So what we are looking at is the metabolic differences between ApoE carriers. So in the brains of ApoE4 carriers, we actually see altered glucose and lipid metabolism. So here in this picture, what we're looking at, the purple is actually patients who have been diagnosed with Alzheimer's disease already. And what we're seeing here is the regions of glucose hypometabolism in their brains. And the blue is actually people from 20 to 39 years old or 47 to 68 years old. And it's the overlap of glucose hypometabolism in their brains already in these ApoE4 carriers as young as 20 years of age. So ApoE4 carriers also have mitochondrial dysfunction and alterations in their pentose phosphate pathway. So once again, just to give a little background on the gut-brain axis. So as we learned, stress and pathologies such as Alzheimer's disease actually cause the gut to be in dysbiosis, which leads to a disrupted barrier. It leads to inflammatory cytokines, an increase in amyloid beta in the brain. It also leads to blood-brain barrier permeability, impaired brain metabolism, and neuroinflammation. And what we want to do is we want to see if we give these mice with this ApoE4 gene prebiotic diet, if we can remediate these things. So we want to see if we can get the gut back in eubiosis and producing neurotransmitters and short-chain fatty acids, as we heard about earlier today, remediate the tryptophan metabolism, change the inflammatory profile, improve brain metabolism overall, and reduce neuroinflammation. So just a background on this prebiotic diet. So as many of you know, a prebiotic diet is a non-digestible food component. It's found in fruits, vegetables, beans. It beneficially modulates the gut microbiota and increases short-chain fatty acid production. Examples of these are resistant starch, and the one we used is actually called inulin. Things like wheat and shallots, leeks, as you can see here on the screen, is where inulin is found in the foods that we eat. And it's well studied in GI disorders and other metabolic disorders such as diabetes, but there's actually not any research on ApoE4 carriers and the effects of inulin on their brains. So we want to know, can inulin restore their metabolic function? So as I just stated, we want to use this prebiotic to enhance metabolism and decrease inflammation, and we hypothesize that it will reduce disease risk for these asymptomatic EFAD carriers. So what I mean by EFAD is the mouse model that we use is actually a human-targeted replacement ApoE mouse. So it has a human version of the ApoE4 gene. It has 5 AD mutations so that these mice can actually form those sticky amyloid beta plaques that we all know to be such a hallmark of this disease. And in our diets, we actually see the prebiotic diet has 8% inulin, and our control diet, we use 8% cellulose. And what we did is we fed these mice from 3 months to 7 months, and then at 8 months we did hippocampal MRS, we did blood and fecal metabolomics, and we did a microbiome analysis. And how I'm going to walk you through these results is actually kind of like a step ladder. So we're going to start with the gut, then move to the blood, and then check out the results in the brain. So as I was just saying, inulin changed the gut microbiome diversity in these E4 FAD mice. So the alpha diversity, which is within a mouse, it actually didn't significantly change, but if you can see here, there's a little bit of a difference between the control and the inulin-fed mice. And actually looking at the beta diversity, which is between mice, the control group and the inulin group have a significantly different microbiome. Inulin here in the blue and control here in the red. Just to get a little bit more in depth into this, we saw an increase in these E4 inulin-fed mice of lactobacillus, which as we all know is a common probiotic, produces beneficial metabolites, inhibits pathogens. And we also saw a decrease in things like Escherichia, which are species that are implicated in causing GI disorders and inflammation. So next we look at short-chain fatty acids in the cecum, and as we learned today, short-chain fatty acids have various beneficial effects. So we saw significant increases in cecum acetate, propionate, and butyrate, and they do differing things. So acetate will decrease appetite, it'll increase mitochondrial efficiency, propionate increases glucose sensitivity, and butyrate increases things like neurogenesis and memory and cognition, and also decreases inflammation. So obviously we're seeing these things in the gut, but are they going to get to the brain? That's obviously the goal of this. So here we see a significant increase in blood acetate, increases in blood propionate and butyrate, but they weren't significant. And then next, in terms of tryptophan metabolism, which plays a role in inflammation, we saw increases in IPA and indole acrylate. So these are both part of the indole pathway, which is known to modulate immune signaling through the AHR pathway, and IPA is also known to inhibit amyloid beta aggregation. We saw increases in kynurenine, and this is a neuroprotective pathway of tryptophan metabolism. It also modulates inflammation, and we also saw increases in serotonin, which is obviously implicated in brain signaling. So we're seeing positive implications for a modification of the inflammatory profile of these mice. And then in terms of the TCA metabolism, so as you remember, these ApoE carriers actually had glucose hypometabolism from a very young age, so this is something that we're really trying to target. Seeing an increase in succinate, fumarate, and malate, which are all parts of the TCA cycle, which as we know are is part of the glucose metabolism, is it's really beneficial. It's really positively modulating the glucose metabolism in these mice, and that's something obviously that we're trying to target. And then we also saw alterations in the pentose phosphate pathway. The pentose phosphate pathway also involved in glucose metabolism, but something else that this pathway does is it generates NADPH, which helps prevent oxidative stress. And obviously this is a huge part of Alzheimer's disease in the brain. So we're seeing increases in blood ribose, ribonate, ribulose, and arabinate. And then in terms of the inflammatory transcriptome, we're seeing significant decreases in things like CCL4, CXCL10, FCGR4, and ITGAX. And these are obviously involved in various parts of the inflammatory pathways. So seeing decreases in these has positive implications for reducing inflammation in these ApoE4 carrying mice. And then the last thing, so sialoinositol is actually produced by the gut microbiota, and it has been used in clinical trials to inhibit amyloid beta aggregation. So our hypothesis was that if we increase inulin, we're going to increase the amount of sialoinositol that's produced by the gut, and this will help decrease amyloid beta in the brain. So what we did is we did hippocampal MRS, and we actually do see a significant increase in sialoinositol in the hippocampus. So we're hoping that we will see amyloid beta decreased in the amount of plaques that are in the brains of these mice. So just to summarize, when these mice were fed inulin, the ApoE4 mice saw an increase in the beneficial gut taxa. They saw an increase in tryptophan metabolism and decrease in neuroinflammation, as well as improved glucose metabolism and an increase in short-chain fatty acids. So we're seeing a slew of positive things happening. And obviously this is mouse study, so it's preliminary evidence, but it's positive preliminary evidence. So just for future directions, we want to continue and do behavioral tests as well as amyloid beta staining in order to see if our hypothesis about the sialoinositol was right. And then we also believe that this study, although it's preliminary, is highly translatable and personalized. So we know that prebiotics are safe for humans. They're already used in humans. And all of the analyses that we do on these mice are readily available for human studies. And then even further in the future, we want to look at ApoE3 carrying and ApoE4 carrying mice and assess the nutrigenetic and nutrigenomic differences on this prebiotic diet. And this has implications not just for Alzheimer's disease, but for other diseases that this gene is involved in. And we hope to be able to develop a personalized nutritional intervention based on ApoE genotype. And lastly, I just want to thank my wonderful mentor, Dr. Eileen Lin. Without her, I wouldn't be here, have the opportunities that I have, and all of my colleagues in the lab, as well as the Department of Nutritional Sciences at the University of Kentucky. Thank you.